Hi, everyone. I'm Heather Gatney, and I'm the coordinator of the CVFS webinar series. Our host today is Coulter Mitchell, Research Associate Professor at the Survey Research Center at the University of Michigan. Today, Coulter is going to tell us about analysis methods using polygenic scores for demographic behavior. There will be a Q&A session after the presentation. We ask that you remain muted until then. During the Q&A, you may raise your hand to ask a question or use the chat. Also, please use the chat during the presentation if you have a clarifying question. We'd also like you to know that this webinar is being recorded and will be available to view from the CVFS website. And we'll put the slides up there too. Okay, Coulter?
All right. Hi, everyone. Hopefully you can see my screen now. I'm getting a couple nods, so that's good. Or you're just not paying attention, one of the two. Yeah, so please feel free to the clarifying questions, jump in with those, and please, if anyone's tracking that, stop me. I'm happy to address things partway through. So I'm excited to talk about genetics and demographic behavior. It's something that I think I have talked about for quite a few years now, but actually never put together a more streamlined version of all of these different things. So I'm excited to talk about it, and in particular in relationship to the Chitwan Valley Family Study, because I think it's exceptionally unique opportunity to study demographic behavior and genetics in particular. Okay, so with that, I'm going to cover a few things. We have an outline. At first, I'll just start with some of the key issues for understanding how we link genetics and demographic behavior. And honestly, we could probably spend the whole time talking about these. I'll then cover very, very briefly kind of what we already know about the genetics of fertility, mortality, migration, and sprinkled throughout those, particularly in the fertility section, will be brief reminders of some genetic analytic techniques. And there are at the very end, I'll point out some prior webinars that we've had on this too. So these other things I won't discuss in detail, but I'll briefly mention, and there's whole other lectures on them. And then we'll talk about the focus on more recent kind of polygenic associations, and I'll describe what that means in a little bit. And then I'll mention again at the end, and there's a whole other presentation on this, CVFS's genetic data. And hopefully throughout the entire thing, we'll be talking about the potential in CVFS and beyond. Okay, so let's talk about the key issues. So let's think about demographic change. I think many of us are population scientists, or if you've worked with CVFS, you know how powerful it is for studying demographic change. But let me reorient you a little bit. Let's talk about demographic change in kind of the idea of genetics and how long of a time frame we're talking about here. So here's just a few examples of how long ago our distant relatives have existed. So you have our cousin genetically, our rhesus macaque and chimpanzees. They diverged from us. We shared an ancestor 25 million years ago. Chimpanzees were 5 million years ago. You have Neanderthals, which are kind of our closest species that aren't modern humans. That was 500,000 years ago that we diverged. My point in saying this is demographic change related to genetics and how we trace it with genetics is a much slower process than you might think. So whenever we think about this, even within most species, the most within species changes require thousands of years, or at least, we'll say 1,000 years before you start to see major differences. We talk about things not in terms of years, but often in terms of generations. So how many hundreds of generations? And for a generation, you're looking at 25 to 30 years. So keep that in mind. Genetics don't change that rapidly. The one exception, that you see a little bit faster are very powerful rare mutations. and we'll talk more about that in a second, that even require many generations or hundreds of years. Oh, good, Erin's here, so she can answer all these questions too. So I've just set up how long it takes for genes to change, for us to see really discernible changes in the population in genes. And now let's just look at some more demographic related things. So let's look at child mortality rate between 1800 and 2022. So about 222 years worth of child mortality in different countries. And what you see is that we had fairly high child mortality until, towards the end of the 1800s in some countries and even towards the middle of the 1900s in others. And now it's rapidly declined. So this is a change over 150 years or maybe even over 100 years. Now let's look at the birth rate. That's the one below that. Again, you see pretty high birth rates in some countries. Again, early 1800s, some are more a steady decline over 200 years, over 150 years, and others have very rapid over, say, 50 years or even 30 years sometimes. So you might say, well, if you're a good demographer, you would say, well, I think I have just figured out that genetics did not cause, let me go back to these two changes. It's kind of impossible, right? So why would we even study it? If the most recent demography change cannot be through genetics, it just doesn't seem possible. Yet, how is it that genetics still influences demographic behaviors? So I think that's actually a fundamental thing to consider when we're talking about this. So I think one thing to know is there's kind of this constant genetic diversity that we'll get into in a little bit. That genetics are related to lower fertility or mortality, something like that. So we have things like rare variants and that just affect kind of the general distribution of fertility and mortality. We also have kind of a new historical environment, right? So you can imagine that in a world in which there's natural fertility, certain genes might play a very big role in determining variation. And yet in a time when it's highly constrained, or maybe timing is very different, maybe new genetic effects. And that's kind of a broad historical by gene environment interaction. The other thing that I actually think is incredibly important for us to think about is that genes are highly correlated with environments. And the reason why that exists is often through different social factors. Certain groups are in certain regions, they do certain things, they're allowed to have certain privileges that allow them to have higher numbers of children or smaller numbers of children. And those genes are then linked to the outcome that we're interested in. But it's through environmental processes, not a genetic biological one, a direct one. And finally, and this is kind of where I think there's a lot that is quite powerful, is that it's really quite easy to highlight social processes with genetics in the ways that I was just describing, that maybe genes appear to have a direct effect on something, but in fact, social scientists can show that it's primarily operating through a selection on some other outcome or a social environment. And I think it's a great way to highlight that, especially considering that there are still a large number of people who doubt that social factors can drive a lot of these, what they might consider biological processes. Okay, so that's one thing to keep in mind when we're doing all of this. And I spent a lot of time on that first part just to set it up. So one thing to keep in mind as well is on the left, you have kind of different effect sizes. We have a chart with effect sizes on the y-axis, so really strong effects. On the x-axis is allele frequency. And what that is just essentially a measure of how rare something is in the population, how rare a genetic measure is. And in general, what we see is that very rare alleles often have very strong effects. And that kind of makes sense, right? So if something is very rare, in other words, it's so rare that if you have it, it's going to make you, um, die of it, then it's probably not going to last very long in the population. Um, and uh, and you can think of there's lots of examples of this, and we found those even before we could do genotyping because it tracked into families. And what we found is that most things, most behaviors that we're interested in actually are uh, not just the effect of one gene or one genetic measure, but actually um, the kind of sum of thousands of very small effects. So they're common. In other words, they're more common in the population, have a higher percentage in the population, but they have very, very tiny effects. So tiny that if you were just to look at it, for one, it wouldn't even show up in a pretty big study. It takes very, very large studies to detect these. The other thing I think I've mentioned already is gene-environment correlation. So keep this in mind. There are different types of gene-environment correlation. I think the thing that people most often think about is active gene environment correlation. That is, people choose or maybe even create an environment based on their genetic tendencies. So if you want to go very extreme, that you have some genetic profile that makes you want to do something more often. We actually find that that doesn't really exist, but that's often the way we talk about genes, right? Like, oh, you have a genetic proclivity for uh, being an extrovert. Therefore, you like going out and meeting new people. You be a great salesman, that sort of thing. That philosophy, that idea is gene environment, active gene environment correlation. More often, I actually think what is actually happening is the other two, which is evocative in gene environment correlation, which is when the environment is responding to a phenotype caused by genes, but they aren't necessarily the gene that you would expect, right? So uh, it may be that you have genes related to, let's make it very obvious, skin pigmentation. And then you get treated by society in a certain way. You get placed into certain things. You are required to do other things. And then that influences your environment, your health, all of your outcomes. And so it looks like there's a genetic link between, say, skin pigmentation and this outcome, when actually there's no direct biological association. It's all operating through the fact that there's a social selection of that person into an environment. So the environment is selecting on something and putting you into a place. And you can think of lots of social processes that operate that way. And then finally, passive gene environment correlation, which again, I think is a very big one. That is that, say, your parents or your ancestors have certain genetic makeups. They also lived in certain environments, and both of those things get passed on for many, many generations. And it's not necessarily active or evocative, although maybe it started that way, but actually it's just that they get passed on together. And again, I think social scientists are very well aware of how that works. The final thing, or I don't, maybe this isn't the final thing, but one of the other things to keep in mind here is when we say things like race or ethnicity or caste, those are socially constructed and key measures in understanding social and health inequalities. We use those all the time. They're very real in their effects, but they are socially constructed. I won't go into that more detail because social scientists usually have talked about that extensively. Um, Ancestry is different. When we talk about ancestry and genetics, what we're really talking about is variation in genetic architecture between populations. That's primarily used in genetic analyses. And what this is really a result of many generations of demographic processes, including migration, fertility, and mortality. In other words, this is really talking about where your genes came from and, uh, and how they're correlated with other populations. Um, but a lot of times these all get mixed together and they really are very two separate things. And sometimes, in some situations, someone's ancestry might be highly correlated with what their race is. For example, in the United States, if someone says that they self-report as white, they very, very often have a very high percentage of European ancestry in their ancestry. But there are many other races that will say they self-report as some race, and their ancestries can be from have a wide range of ancestries in them. So it's not a perfect correlation, even though it often gets linked that way. Just to give you a sense of what this is interesting, this is one of my favorite genetic studies ever. This was looking at, this was a study a while ago at this point that just took families that had lived in Europe for at least 3 generations. And then they took the first 2 principal components of all their genetic data and they rotated it a little bit. And lo and behold, it maps onto what the European continent looks like. So you have quite literally people who, like in Portugal and Spain, kind of cluster together, right? Italy is closer to Spain than it is, say, to Russia, but it's still on a different axis, and it actually really does line up quite well this way. And in fact, a lot of genetic studies, it's very easy to separate out geographic populations because their genes are actually highly correlated with each other with the people who they've been living next to for many, many generations. So visually, genes kind of map onto our history of migration. And we'll talk more about that in a little bit. Okay, so now we're going to start into, well, before I go there, if there are any questions, please let me know. Okay. Jump in at any point. Now let's talk a little bit about fertility. So if you dive into the genetics of fertility, the reality is that a lot of the work, and it's a very big literature, really focuses on the genetics of infertility first. And what you have there is there are different estimates. These are within genetic studies. There are other ones that are more social focused, but they'll find that around 7% of couples who are trying to have children cannot have children. And there's actually a relatively large literature that has looked at this, and it's not surprising. This gets back to those rare variants. There are some genetic effects like chromosomal anomalies, XXY, XXX, different microdeletions on the X and the Y chromosomes. Those are the sex chromosomes that it just takes that one mutation or that additional chromosome, and you have it's almost impossible to have children. Although it happens rarely, but it's typically impossible. And there are over 20 single genes that, and they're typically different by sex, that have effects on infertility. And in fact, if you're going for treatment or things like that, they'll often do a genetic test, and that's how strong these are. If you have it, then they have to think about other options. And that literature is very large, it's strong, but as I said, it's actually quite rare. And so it has a large effect, but on a few percentages, a small percentage of the population. And one of the things that's kind of happening right now is that they are discovering kind of newer genes, sets of genes, in some places where there's been a lot longer delay in fertility. So this is like age at first birth kind of effects. And they're seeing new genes that they didn't see before because people are delaying longer and they wouldn't have had that opportunity before. But what I want to focus more on is polygenic exploration. So this is more common variants, as I was describing before, that have kind of a broader effect on, say, the number of children or the timing of children ever born. So what this comes from is high throughput techniques. That just means that we can get a lot of data relatively quickly, have allowed for more genome-wide analyses. So not just a targeted single gene or a single chromosome analysis, but across the entire genome. So this is where we get into genome-wide association studies, and I'll talk about that briefly on the next slide. Identifying genetic variants associated with complex traits, in this case, fertility, by scanning a strategic subsample of all of your possible genetic variants. just so you know, most of the time when we talk about genome-wide association studies, we're talking about, we haven't measured the entire genome. That's called sequencing. This is typically a sample of the genome, targeted, but still a sample. And fertility outcomes such as the number of children ever born, age at first birth, age of menarche or menopause, which I won't be talking about those, are all influenced by both genetic and environmental factors. So here's just briefly how a genome-wide analysis typically works. You start with a discovery sample, and this is very simple. Ordinary least squares or logistic regression done millions of times. So we might have 5, 10, 11, 25 million genetic measures that we have to test, and we'll do that. And we'll use, to account for kind of the multiple testing, we'll have a p-value that's not 0.05, it's 5 times 10 to the negative 8. And we take anything that's kind of more significant than that, so smaller p-value. And there's various clustering techniques and correlated markers that I won't get into. And this is typically done in large meta-analyses. So this isn't something that one study can do. This is typically over many studies. And we're talking about hundreds of thousands, if not even millions of people. So you're combining a lot of studies that were collected in many different ways that may have slightly different ways that they've measured things. The idea is that you're going to combine all of them. They've all been genotyped. And that's a little bit more rigid in the way that it's been measured. And then if they find anything, they then have to take it to a replication sample, which is still a smaller n, but typically still rather large, still often tens of thousands, if not hundreds of thousands. And again, if they have multiple cohorts, they'll do a meta-analysis of that. So that's how a GWAS works. This is what the results look like. I'll show a few more of these. This is why I'm explaining it. So on the x-axis, you have essentially the address of the chromosome, and on the y-axis, you have the p-values. So the higher it is, the more zeros there are before that 0.01. So smaller p-value. And essentially what you're looking for are these peaks. These peaks are, because there's highly correlated genetic measures right next to each other, you'll see this rise of hits, what we would call them, anything over that line. And that's where we say, oh, it looks like there is a signal there that we should investigate more. So all those red circles, that's what that is. No questions yet. Okay, so let's look at an example of a fertility GWAS, just for fun. So this is not the most recent one or the biggest one, but it's, I think, a particularly good one. And I have some other examples of other ones in the slides. I try to put the key fertility and mortality GWASs out there so you can kind of look in depth more if you want to. But this is a good one. So this identified 12 new loci. That just means 12 locations. In this case, they're mostly genes influencing human reproduction. So they studied age at first birth and number of children ever born. But as is the case with most genetic analyses, it's a group of multiple cohorts. In this case, almost all of them are European ancestry cohorts or ancestries. And that's the vast majority of this work. And in fact, most genetic work is done on European ancestries only. Although that's beginning to change, it's still rather slow, in part because we just don't have a lot of other cohorts, not nearly as many proportionally. But you can see we had 250,000 for age at first birth and 343,000 people for number of children ever born. And so I said that they had 12 loci. These were 10 new gene regions that were discovered. 2 had already been found. So if you go here, now you are all experts in looking at a Manhattan plot is what this is called, because it's supposed to look like the skyline of Manhattan, but you probably have other cities that we could describe too. But again, chromosome is on the x-axis, so that's just the address. And on the y-axis is the p-value, where the higher one is a smaller p-value. And again, these genes don't necessarily mean anything to you. That's what the paper goes into, is what each of these genes does. But you can see those signals of those 12 different loci that found something related to, say, age at first birth or number of children ever born. A lot more on the age at first birth. In this case, that was on the left. The other really interesting thing is they can take all of those hits and they can say, well, we have huge databases now that compare the things that we see for, say, age at first birth. Do we see anything that those same sites, those same loci, link to anything else? And in this case, you can actually do a genetic correlation. So are the genes found in one thing the same genes as found in some other trait? And this is a fun graph. I won't go through the whole thing, but it just starts to show like, oh, if I look at genes related to age at menarche, I see that it is positively correlated and significantly correlated with age at first birth, which makes sense. Higher age at menarche, higher age at first birth. Or you look at something for number of children ever born, which is the blue bar, and you see that number of children ever born, the genes related to that is negatively correlated with age at first sexual intercourse, so genes related to that. And you can kind of go through and see which ones are significant, which ones are not, and how they're correlated or not correlated. But these are kind of the cool things you can do with genetics. And so actually this comes from in part from something that Aaron Ware, who's on, wrote a great paper describing how you can actually take all of that information that I just showed you and create a score from it. So the paper's down there in the left-hand corner. So what that is imagine that someone has written a paper that has found, say, major hits, major loci related to fertility or number of children ever born, something that you're interested in, and it wasn't done in your sample, right? It's done outside. What you can do is you can take all of those results, and because we know the addresses, you can pull all that information, and what you do is then you extract that and you can create a score. That score is a combination of the weight. In this case, it's usually like the effect size or the inverse of the p-value. So something that gives it kind of the, how important that genetic measure is, and you multiply it by whatever the person's gene was in your study. And you can add all of those up across some number or all of the genetic measures that you have. So 25 million, 11 million, 5 million, however many you have measured. And you can combine those and create a score for each person that's, say, predictive of whatever outcome. In this case, let's say number of children ever born or age at first birth. And so people have done this. They take those outside or external studies, the GWASs, and they create scores and they say, let's test it in my study. And in this case, that might make sense. However, one of the things that's problematic within genetics is that the polygenic scores that we just talked about creating have some challenges being portable by ancestry. So if you remember correctly, we said that the first study that we looked at was mostly European ancestry. It may not work for other ancestry groups quite as well. It still is often predictive, but at a much lower rate or much lower effect. So let me just give you an example from one of the studies that I've done. So this is looking at different height polygenic score for height in a population of children at age 9, I think. I think this is age 9 or age 15 in the United States. And there you would see like, oh, the African ancestry population is we would expect to be significantly shorter than the European, which is the yellow, and Hispanic would be between the two. And in fact, when you start estimating how much it would be, it's like European ancestry would have expected to be 4.3 inches taller than African ancestry. And so you're like, oh, well, that seems strange because we actually have measured height and it's not that different. And that's because the European ancestry score works very well in European ancestry. It just doesn't predict nearly as well. Still predicts, but it's a much weaker association in other ancestries. And we kind of see this repeatedly for almost all traits. Again, they're getting better at making them more portable, but it's always a thing to be cautious of and just be aware of that if you're taking something from one ancestry group and predicting it in another, it's probably not going to predict quite as well. But people have done this. They've taken polygenic scores of fertility and tested them and say they created it in one, and then they tested it trying to predict variance of age at first birth and number of children ever born in other studies. And the figure on the right comes from this study, like in the Health and Retirement Study or Swedish Twin Registry, how much of the variance was predicted. And this the x-axis in this case is about how many CPGs or how many genetic measures or markers they used. That's not as important, but you can see how much variance it's explaining. And like a lot of things, these, even when added all together, it's still a relatively small amount of variance being explained. You start getting up into the 1%, and there's some other scores that get up into 3%. So it's not a huge percentage, but it's also something. And the thing is, you can take that and you can say, I want to predict fertility, but maybe you also want to see, is it associated with other traits? Like there's a study that looked at how scores related to fertility were related to extroversion, or others that were related to externalizing behaviors for risk-taking, menopause timing, puberty, obesity. And so there's a lot of trying to sort out how these genes are resulting in fertility outcomes. So what are the mechanisms? The other thing that's really powerful is you can take these scores and by having, say, things like siblings, you can start to say, okay, it's random which sibling gets which set of genes from their parents. So, so comparing two siblings who have slightly different scores, that difference you can kind of assume as a random, and you can start to test, is this actually a causal association between these polygenic scores? But as I'll say before, I think there needs to be a lot more work in non-European studies, which Chitwan Valley Family Study certainly counts as. Longer kind of developmental, or in this case, I mean like child development, or life course approaches to studying this, and of course, how it's correlated with the environment. Okay, so that's fertility. I know I went through that relatively quickly, and these next two are going to be even faster, just so we have time to talk. I started with fertility. In some ways, it's the cleanest one, and also it's the first thing that we always say is fertility. So I'm going to go to the end now, mortality. So again, there's been a lot on the genetics of mortality and lifespan. And it's also influenced by a complex interplay of genetic and environmental lifestyle factors. There have been large-scale GWASs to identify things related to mortality and lifespan. You have notable genes like ApoE or FOXO3 that were found very, very quickly, and in fact, kind of challenged our genetic studies because we found them so quickly right at the very beginning that we kind of assumed all of the things that we studied were going to look like that, and we just happened to get lucky and found the biggest effects right off the bat. And then most of them are not that strong. But these genetic predispositions can partly explain variations in lifespan across populations. So some populations do seem to live longer in part because of these genetic predispositions. Okay, so here's an example of a study again, GWAS of lifespan in this case. And when they did this study, they found 25 new genes related to lifespan that they hadn't found before. So this is a study in 2017 on 389,166 UK biobank participants. So already you probably know a lot about what that sample looks like. So UK biobank is very homogeneous, European ancestry predominantly, and it's not even just all of Europe. It's one relatively narrow part of Europe. So European descent. UK biobank is not necessarily generalizable. It has a very large sample size, but it had a response rate of 5%. So they were caring more about getting people in than necessarily recruiting only the 500,000 that they tried to get, for example. And in this case, what they did is they looked at data recorded on parents' current ages or their parents' age of death, and they used the child's genes and asked about their parents. And in this case, they had around 181,000 who had one or more living parents of the 389,000. So many had parents who died. So they used kind of a triangulation method of having highly related phenotypes or outcomes. So they had the combined age of the parents if they were living, the combined age of death if they had died, if the parents had lived to at least the 90th percentile, which is age 90 for women, age 87 for men. And they just, like I said, they discovered several new loci for biological exploration. And they took those polygenic scores, which I won't go into detail. They took polygenic scores from those and took them into other studies and predicted mortality in multiple longitudinal cohorts. So this does produce a lifespan score that is predictive of how long people might live in other studies, even non-European studies. So again, this is what that GWAS looks like. In this case, you had that combined age of death, and parents who lived over 90%. And again, now you're starting to get used to these. You have the x-axis, which is the chromosome address, and the y-axis, that's the p-value. And you see, again, these trails. And you actually kind of look down A, B, C. You see similar ones. And I'm going to take one that's kind of an interesting thing to do. So I took one, let's look at this one on chromosome 9. And we can actually do, we can zoom in on one locus and kind of look at some of these. So at the top here, you see that top hit, what we would call it, for parents' attained age. So that is one genetic measure. And you start to see, oh, here's some other ones that are highly correlated. That's what the colors mean. The red means they're highly correlated with it. And in fact, those had a lot of them had been found to be related to different like strokes and aneurysms and MI. So that makes sense. Those are ways in which people might die. It's moderately correlated with other survival or cancer genes and other health outcomes. So you can start to see like, oh, that makes sense. This one is now known related to longevity, but that's probably because it's also doing things related to, say, cardiovascular function or something to do with brain function or cancer. And so this is how we do those biological explorations. The other thing to know is that even bigger than lifespan or mortality analyses, there's a huge number of studies just related to specific disease analyses, whether or not it's just getting disease or mortality from that disease. In fact, so much more related to, say, cancer or neurological diseases like AD or cardiovascular diseases. In fact, there's entire institutes devoted to each of these. So it wouldn't necessarily be mortality more generally or lifespan, but it would be mortality related to cancer. That's like the National Cancer Institute spends a lot of effort trying to discover that. So here's some examples of other ones that you could look into. Some papers down there. So there's a lot of promise. The reason why people are spending a lot of time, advancements in genetics hold promise for targeted therapies or personalized medicine approaches to enhance longevity. And this is where I'll say, take a step back and say, but also there is a strong potential to lead to health disparities unless the data, the research is more representative. And there's ethical considerations to include potential discrimination based on genetic predispositions for like life insurance and things like that. And there are protections in some countries, not in all. And that last article, Alicia Martin's, that I have listed on there talks a lot more about the potential issues about using these, even though there's a lot of promise there, because it's not necessarily representative yet, some potential for strong potential for health disparities to emerge if we start using those. Okay, so migration. I'm going to move to this one because I want to make sure we have time to discuss some other ideas. the interesting thing about migration is that in some ways, you already heard me talk about this. It's been the primary thing that has been studied with genetics. Genetics reveals historical migration patterns. That's what it's actually probably most well known for doing before we started getting into these high throughput, more recent, like last 15 years or so, research. Before that, it was all about how long ago someone left a certain region or who they migrated with. So it's a key factor in human evolution, shaping genetic diversity and population structure. The majority of genetic studies of migration investigate historical movements of populations. There's been some recent advancement in genomic technologies that enable kind of more detailed patterns. And again, keep in mind the timeline I'm talking about. Recent migrations are like the last 1,000 years. Um, and even those continue to impact genetic diversity. So we're not talking about migrations in the last 100 years changing things. We haven't seen the evidence of that yet. And the focus here is descriptive, it's not causal. So here are some major migrations inferred through genetic. And again, I can't spend a ton of time on this. I wish I could, but it starts to show like how long ago, just using genes, not using archaeological evidence, not using anything else, just using genes, where they've been able to tease out how long ago did populations leave Africa. That's 55 to 65,000 years ago. At what point did they end up, say, in what we now would call Australia? That was 47.5 to 55,000 years ago based on genetics. And then you think about Neolithic expansion, that's up here, that's 10,000 years ago. But like the northern southern split in North America was 13,000 years ago. All of those things have been described just using genetics. And so it's fascinating, but it's highly descriptive. And people have studied this too, and environmental selection. So migration has facilitated some evolutionary adaptations, say living in a very high altitude and selected on certain genes related to that. Like high altitude diet, infectious disease are certainly historical migration patterns. But again, you have to think about several generations for this to manifest. This is not an instantaneous thing. So we won't have a COVID-19 related, genetic patterning for a lot longer, if at all. So there's much less focus on what genetic traits lead to migration. I'll show you one example of this that I find interesting. And really no focus on whether or not it's evocative versus active gene environment selection. So in some ways, this kind of gets into the forced migration versus, chosen migration processes. I've never seen anything really documenting that. So there's a lot of potential for exploration of genetics of different traits predicting migration. It might be difficult, though, to conduct a GWAS of migration without a lot more non-European data, or we might have to limit what we mean by migration. Is it international migration? Is it within region? What counts as migration will be helpful to think about, but it hasn't really been done in the same way. Here's one example from the Dunedin study, which is in New Zealand, where they just created a score of education, a genetic score of education, just predicting how far you would go in school. That was what it was designed for. But then they ended up showing that those genes also predicted what type of partner you married. So that's kind of related to fertility often. And then they also showed that it was related to migration patterns. Who left? So if you had a score for higher education, remember this is something you can measure at birth, they were also more likely to migrate internationally and even differences in where they would end up. So it's interesting, but a lot more work, so much more work to be done in this area. Okay, so let me just kind of wrap this up by talking about the Chitwan Valley Family Study, genetic data, why I think it's so powerful for studying this. So I just want to show the CD data, and actually there's already been a couple talks on this, so I won't go into too much detail, but the reason why that's important is this is also when the genetic data was collected. So just keep that in mind. It's tied to that data set. And Bill, please correct me if I say anything incorrect at this point. but this is what that looks like. It has a nice description of the whole population. And keep in mind, that's what the genetic data will look very similar to, because we used saliva. It was easy to collect. And 96% of the participants provided saliva, which is an impressively high. In fact, I don't think I've ever seen anything higher for biological data participation. And there's different consents for DNA collection and sharing data. Just so you have a sense, and I'll show another version of this, so you don't have to remember all this, is how many different individuals there were, around 10,000. Were they located in trios, like mother, father, child? More than half were, but also there's one parent or one sibling, greater than one sibling pair, but maybe not a trio, a parent-child duo, no parents, but had a sibling comparison, and totally unrelated from everyone else in the population around 1900. So this is the part that is both incredibly exciting for me and for you, and we can talk more about this. Is what I'm kind of hoping we spend more time thinking about, are the strengths of the study. As you know, and there are already several talks on this, you have prospectively measured demographic behavior in highly well-timed measures. It's not like I asked someone at baseline and then I asked them 20 years later. Very precise measures of demographic behavior, fertility, migration patterns, and mortality too. Strong focus, incredibly strong focus on fertility and migration. Some of the best measures on fertility and migration that we could have. Large sample for testing polygenic scores. Keep in mind, again, there might be that issue of if it's coming from a European ancestry, it may or may not predict as well. The use for trios, mother, father, child combination. In this case, we think we have around 2,000 total trios. It does help with breaking some of that, what I would call population stratification. That's related to that ancestry differences. So that can help. There are some ways that you use trios particularly uniquely. There's also this thing called genetic nurturance, which I'll spend a little bit more time talking about. This is the concept that parents' genes affect the outcome of a child both directly and indirectly. Clearly, it makes sense. Parents' genes are directly given. For some of those genes, about half of them, they're given to the child, right? And so those child have those genes, and then that means that they might act a certain way, they might be a certain way, they might achieve certain things. So that's the direct effect or not direct, that's directly to the child that they've given it. And then that's actually indirect to the outcome, but there's also the process of the parent's genes influencing, say, the way they parent or the selection into certain environments. And that can have a direct effect from the parent to the behavior. And so there's this whole world of trying to tease out to what degree parents' genotypes influence children's outcomes through giving their own genes to them or through the creation of an environment that then influences it. So I think there's a lot of work to be done here. It's a very exciting time, and there aren't very many studies that can do this right now. And in this case, we have over 5,000 mother-child pairs and 4,500 father-child pairs. And then finally, I think as we talked about before, the sibling comparison. So around 2,000 to kind of estimate that causal differences, right? So if you have two siblings, they, in theory, were given the same chances to receive the same polygenic scores, and it's random which one had a higher or lower score. Okay, so I want to come back to this slide. I just, at the end, just note there's several prior lectures that I think might be really helpful at introduction, things related to migration, to health, genetic designs and methods. So please go back and look those if any of these things is important. I also left some slides for links if you're interested in social scientists studying genetics more broadly, the NIA Biomarker Network, courses for genomics for social scientists, and other consortia. And that's all I have, but I'm going to go back to the strengths just because I think this is where I want to talk more about things. And I'm done.
Thanks so much, Coulter. And you leaned into the demographic outcomes. Always appreciate that as a demographer and what we asked you to do. I note that some of our participants are also interested in psychological, psychiatric outcomes. So I'm going to make a brief comment about that. Coulter pointed out that the sample originally was what he called the CD. That stands for Composite International Diagnostic Interview. That's the World Mental Health Survey's Composite International Diagnostic Interview. And we worked with a team of psychiatric geneticists at Harvard who are still working with these data. Our first publication, which uses polygenic risk scores, as Coulter mentioned, to predict depression in the Nepal sample, was published in 2024. If you go to the CVFS publications website tab, you'll find that reference there and you can chase it down. I'll say a little bit different. Coulter, I always appreciate your emphasis on the bias that comes from having European ancestry training of these kind of polygenic risk score models. I'll say that the team at Harvard was careful to use Asian biobank data to try and train that polygenic risk score to be not so Eurocentric. And I'll say that nevertheless, Asia turns out to be big. And as a demographer, I'll say that's because they have way more people than every other region in the world and quite diverse. And so it's not at all clear that the predominantly East Asian genetics in that kind of resource matches with South Asian genetics, and even South Asian genetics could be quite diverse. And part of the reason that the Chitwan Valley Family Study was funded by NIMH to get these data is because there is so little psychiatric epidemiology with genetics from South Asia. Last word on that. Coulter, I don't know the right words for the graphs, but you have the one with the little dots that, like, there's a graph and there's all these genes and most of them nothing, and then there's one set of dots that go way up. The psychiatric genetics team at Harvard has recently completed their initial analysis of alcohol use disorder and found a single thing in the in the genome that predicts alcohol use disorder really well. And there is such a thing for European genetic analyses as well. And it turns out it's not the same one as predicts alcohol use disorder in Nepal. So they're working on that. But that's the current state of affairs with the psychiatric genetics team. And
Great, thanks, Bill. And that is, yeah, a Manhattan plot is
thank
what
you.
Something like this.
Yeah, with all the little dots. Yeah, so it turns
all the
out
little
that in
dots.
alcohol use disorder, it's all flat except for one spot.
Except for one, yeah.
Yeah.
Yeah, so not really Manhattan, more like maybe I don't know.
More like Ann Arbor in that
Yeah,
sense.
what, just flat and then happens to have one gigantic skyscraper.
Yeah. Ann Arbor, at least in the old days. So, friends, we invite your questions, your thoughts, your discussion items. We have a few minutes left and happy to spend our time that way.
Yeah. And if you don't have questions right now, you can always, on the first page, I think my email is on there, so you can always ask things later on. Yeah, it's a really exciting study. Bill, can I say a little bit more about, I know the limited amount about, this was funded by NIH. The genetic data have been deposited, but I will say that it takes a long time. I've had a lot of experience trying to deposit data. There are certain archives that really handle genetic data, and that's their primary thing. It can take years, even after you've cleaned the data, you've prepared it, and you've submitted it, for it to be fully released. That's not the fault of the study. And I think that, as far as I know, that process has started with Chitwan, and it just takes a long time for it to get out. But I don't know, Bill, if you wanted to add anything to it.
Yeah, I'll say that the National Institute of Mental Health at NIH funded this research, and we did deposit our genetic data in their shared repository successfully. Heather, I think you said as of yesterday, it didn't show how to get the data yet, but it's there and they're processing. It should be available. But I'll say that really is constrained to the mental health outcomes of interest to that institute. It's not everything in the CVFS, and I'll comment that, while I agree with you about public distribution, there'll always be constraints on the genetic data because it could be used to identify individuals. And so, if you're interested in using the data, we're here to help you figure out how to get that done. And we have the data and would love people to be using it. Maria, thanks for your comment. Cool that you were in Chitwan and did some work there, and I'm glad we were able to tell you more about the CVFS, and thanks, Coulter, for commenting on some of the other webinars. Encourage you to have a look or contact us.
Yeah, thanks, Bill, for clarifying that. Yeah, it's helpful.
And Heather, I think we can also invite people to send questions or comments or queries to the CVFS website email address. Also, we monitor that and we'll make sure it gets to the right person and that kind of thing. So, we love that folks joined us today and encourage folks to use the data. If we can help you do that, let us know how.
Right, we have a contact us page on the CVFS website, so I think that's the easiest way to follow up with us. And I can add that URL to the chat.
Thank you.
Great, thank you.
Are there any other questions or comments? Okay, well, thank you so much, Coulter, for presenting today, and thank you everyone for joining us.
Thank you.
Agree with both. Thank you.